Hi Science friends. Today we are starting our next mini unit. So it's a pretty short unit. It's only got about 4 or 5 lessons in it, but it's on viruses, bacteria, and biotechnology. So there's not a ton of content, but there is a lot of labs. So I'm glad that we're back in person and we can get to do those. So today we're talking about viruses. So what you need to know about viruses is that viruses are generally considered non-living. It's an infectious particle. It's wrapped up in a protein coat. You kind of see this here. It has these little surface markers. Different types of viruses have different types of surface markers, but that's what your body generally is going to help to recognize it, that it's foreign, and try to fight it. But they are non-living. Now, we generally consider them to be non-living because viruses cannot live without a host. Viruses are only going to last like on a doorknob or on a surface or something like that for roughly 24 hours or so. After that, they are going to have a host to help them, replicate, and so on and so forth. So what viruses do is they inject their DNA or their RNA. Viruses can be classified as a DNA or RNA virus. And one thing that's kind of random or unique about this is they can have single-stranded DNA and double-stranded RNA. So typically we learn that DNA is double-stranded and RNA is single-stranded, but that rule doesn't really apply to viruses. So they can have single or double-stranded DNA or RNA. But inside this protein coat that they have their genetic material, and they basically inject that into the cell and take over the cellular machinery of their host. And that allows them to replicate and produce more viruses. So viruses are smaller and simpler than bacteria. They're gonna, like the smallest virus only has 4 genes, so it's very simple. But some of them would have maybe up to 1,000 genes or so. But you can't see them under a microscope like you can with many bacteria. So let's talk just a minute about how viruses were discovered. They were first discovered in the late 1880s or so, and they were looking at a disease of plants called the tobacco mosaic disease. I don't know if you can see this here, but it's got kind of a mottled mosaic appearance. Maybe you can see it there if I've zoomed in a little. On the plants, they just kind of get spotty looking. And so they were trying to figure out what was causing this. It kind of stunts the growth in the tobacco plants. And so what they did is they extracted the sap from the tobacco plant and they passed it through this filter that was known to trap bacteria. It was a very fine filter known to be able to trap bacteria. And then they took that filtered sap and rubbed it on a healthy plant. Well, what they found out was that healthy plant got infected as well. So whatever it was not trapped by that bacterial filter. And so originally they thought, well, maybe this is just a smaller bacteria than we've ever seen. But eventually they realized, no, that it's not. There are particles that are even smaller and simpler than bacteria. They're not grown on nutrient media, which we'll talk about that on Monday when we grow some bacteria. And they named them a virus. And so that's how they first discovered viruses. So when we look at viruses are held in that protein coat. And that protein coat is called a capsid. That's an important term. So viruses differ in shapes. This is like a rod virus. So they have all different types of shapes. This one's going to look more like the coronavirus, similar to that with those proteins on the surface. This virus is a specific type called a bacteriophage, or just a phage in general. And so a phage is a virus that infects bacteria, and they have a very elaborate, specific shape. And so these proteins that build the capsid, they're called capsomeres, and they help to protect that viral DNA. So, here we have an RNA inside. This one has DNA, RNA, DNA. So it varies from virus to virus, but they hold that in there. There's no other organelles like the mitochondria and the ribosome and those types of things. It's just basically a packaging of genetic material, okay? Some viruses will carry enzymes inside their capsids, but they're not going to have a whole lot of other things, okay? Now, let's talk about the host range. The host range is the number of species that a virus can infect. So some viruses are very specific. They can only infect one host. For example, measles. Measles is a virus that can only infect humans. You don't see dogs with measles or any other animals with measles. It's very specific. It has a limited host range. Some virus, though, like the West Nile virus, we usually associate that with like mosquito bites, but can actually infect horses, birds. Humans can get the West Nile virus that's transmitted from mosquitoes, and so it has a larger host range. So viruses begin when they invade the cell, okay, and then they take that viral DNA and they use it to be able to copy their own particles, okay, and then those particles are released from the cell, and that's how the virus replicates. So the host is going to give all of the machinery to be able to do that. All that DNA replication and transcription and translation that we've talked about, all of those enzymes that do that, that's what's going to be used to replicate the virus as well, okay? So it's kind of tricky in that it can evade detection by the host and get inside and take over that, that machinery, okay? So viruses reproduce in one of two ways. The first is called the lytic cycle, and we'll talk about that one first, and the second is the lysogenic cycle. So the lytic cycle is going to result in death of the host cell, okay? Organisms that reproduce, or viruses that viruses that reproduce by the lytic cycle are called virulent phage. Only going to reproduce by the lytic cycle. And so these type of viruses have a special type of enzymes called restriction enzymes, and their job is to cut. They cut up the DNA of the host cell, okay? So here's this phage here, all right? It's got its DNA up here, and then here is our host cell. So this is a virus. This is actually a bacteria that it's going to infect. Okay, we notice it's bacteria because it's got a single chromosome here. And so that virus integrates its DNA, we see it coming in here, into the host, okay? And then it's going to use restriction enzymes to cut up that host DNA. And you see that done there. It's already cut up. It's not in this nice circle anymore. Okay, so that DNA is not going to do anything now. And so instead, the viral DNA is going to be replicated. And so you see all of these new parts being made, the proteins and all of the machinery. And so as a result, those new viruses will be assembled and then released. And as they're released, you will see that it causes the cell to burst. And so that's why it has the death to the cell in the lytic cycle, okay? So the restriction enzymes are here that cut it up and then take over the machinery and kill the cell, okay? And release lots of new phage that can go out and infect other cells. So just from this one cell, we've made, 5 or 6 more viruses, all right? Now, the other way that they can replicate is called the lysogenic cycle. So the lysogenic cycle generally doesn't destroy the host. So some phages can replicate by the lytic cycle or the lysogenic cycle, and those are referred to as temperate phage. So let's take a look here at the lysogenic cycle. Lysogenic cycle, we're just going to focus on this half. So what happens in the lysogenic cycle is, although we're going to kind of start over here, you have your phage, and the phage binds to the bacteria and it injects its DNA molecule. Okay, so we have the bacterial chromosome here and then the viral DNA here. All right. Now, this viral DNA is going to ultimately be embedded into the bacterial DNA. And so every time the bacteria divides and reproduces, it's going to also copy that viral DNA. And so it's going to reproduce and makes a population of bacteria that are infected by that phage. Okay? So not only has the bacteria reproduced, but the virus has reproduced as well. And generally, the genes, the blue genes, the phage genes that are in there are going to prevent the coding of these other genes. And so that's why it's going to cause, that's why you see some of the symptoms you do. Your normal genes aren't being coded for. Your cellular machinery has been taken over. You've got cells that are dying. And so that's why we see some of the symptoms that we see when we have a viral infection. Okay? Now, sometimes a lysogenic cycle will flip over and go to a lytic cycle, and vice versa. Those can kind of hop back and forth. Okay? So that is all for today's lesson. I hope you learned a little bit there. You have one assignment that I want you to work on this weekend. It's in the back of your binder handouts that I gave you yesterday, and it's a biotechnology survey. So I just want you to say whether you agree or disagree with any of those items. I think there's about 25 items. Do you think genetically modified food is okay or not okay? Do you think cloning is okay or not okay? And it's very specific as to what it's asking. So I want you to do it for yourself. I want you to ask one parent or guardian, and then if you want to do a second parent, perhaps a grandparent, preferably somebody in your family that you can ask as your second person as well. So work on that this weekend, and then if you read at the paragraph at the top, it says to kind of write a reflection and see how your answers compared with your family answers. Maybe you were really right on the same page, maybe you differed from your family, but write a little bit of a reflection on that. So I will collect those on Monday. Okay? That's all for today, guys. Have a great day and a great weekend.